Hello, everyone. Thank you very much for joining this workshop. Today, I'll show you how you can make your own code connections cluster, and it's inspired by the Russian APT map research we've done a few months ago. Before we start, I would like to introduce myself. My name is Ari. I used to be a security researcher and the head of the IDF CERT team, the incident response team of the IDF. My team and I took care of every nation state attack we targeted the IDF, and we had a lot of work to do, as you can imagine. I'm specializing in malware analysis, incident response, forensics, and reverse engineering. And today I'm the VP of research at Intezer. This session was built for government certs based on my experience and the knowledge we have here in the company. So yeah, let's start. And I would like to start with mentioning a few of the unique challenges that government cert teams have. And we split it to three different steps. First of all, the forensics, where you have tons of alerts and you need to find the needle in a haystack, the true positive alert between all the false positive ones. This is a very hard task to do. Now, sometimes you have huge media to analyze, for example, hard disks or memory dumps, which you need to investigate, and it could take a lot of time. Now, the second step, called malware analysis, once you have few suspicious files, if you want to investigate them and spend time on understanding the classification and attribution of the file, this could take a lot of your resources. If you execute a file in Sandbox, the report could be vague and not so clear. For example, it's malicious 5 out of 10. Okay, but what should I do with it? How should I prioritize this specific alert? And compared to other alerts you have as national certs. And of course, reverse engineering process is expensive and takes too long. Could take days to analyze a whole file properly. Now, even once you analyze the file and you know that it's malicious, now you want to block it and track it. Doing it based on IOCs like hash or IP are not the best way to do so, since they're not that effective. For the attackers, it's easy to just change the hash of the file or create another C2 server. Moreover, tracking adversaries and hunt for new samples is not an easy task. And this is part of the things that government certs should do on a daily basis. Now for that, and based on the knowledge we have in the company, we've built a technology that helps the defenders gain back the advantage from the attackers. We call it genetic software mapping. And basically we can determine if a file is good or bad based on the origins of the file, the actual assembly lines within the file. Now, technically speaking, we can take any binary file, disassemble it with our own disassemblers that we developed, and split it to small fragments of assembly instructions that we called genes. Now, with that, we've built a huge genome database that contains billions of those genes, both from trusted software, but also from malicious files. And for every gene, we know exactly where have we seen it, how many times we've seen it before, who's the vendor who wrote it, and so on and so forth. So for example, we have trusted genes of Adobe and Ubuntu, but we also have malicious genes for Tula and Lazarus, which are Russian and North Korean APT families. Now with that, once we have an unknown file, which we want to investigate during an incident, we can run it in this engine, extract the genes, and then compare them to the other genes in our genome database based on the the matched genes, we can determine if the file is good or bad. So for example, once you analyze this file in the genetic software mapping engine, after a few seconds, we can show you that this code is code from Red Hat Enterprise Linux. That's why it's marked as trusted. We can mark this code as code which is malicious. We've seen it in Mirai. This blue code here is what we called common. It means that it's not the pure DNA of the file, and we've seen it in other applications. So this could be some kind of library, for example. And lastly, and this one is relevant specifically for national certs and organizations that are worried from APT attacks. This code, we can mark the code that was never seen before in the genome database. And this is interesting for itself because the genome database is so wide. So if you see a code that was never seen before in the database, this should be interesting, and I suggest to start analyze this part specifically. So you can think of it as if you have an army of reverse engineers analyzing each and every part of code within your organization. This could be Windows file, Linux file, memory dump, different architectures, x86, x64, ARM, and so on. And this army of reversals gives you deep insights on what's going on within every file you analyze, and within seconds. So this is the technology. Now, the question is, what can you do with genetic software mapping? So I mentioned here a few use cases. Of course, we will focus on the third one in this workshop, on creating a code connections cluster. But I would like to quickly show you the other use cases and how you can gain those using the technology. So let's start with quick malware classification and attribution. This is the example that I wanted to show you. And for this file in Intezer Analyze interface, you can see that after analysis, 81, almost 82% of the code was seen before in the Lazarus family. Now, in case you're not familiar with that family, Lazarus is a North Korean family that is being active more than 10 years now. And you can see the other samples that share code with the sample I just analyzed. So this is very interesting, and you can also see the shared assembly code between the file I analyzed and the Lazarus family. And the reason I chose to show this specific example is because when we detected this file, it has only one detection in VirusTotal. Now it's very well detected. But originally, it had only one detection back in June. But the code reuse never lies. So you can see here that we were able to find connections between this sample I analyzed and previous Lazarus samples that are dated back way back than June, for example, April 1st. So at the first time someone uploaded this file to Intezer Analyze, this APT file, although only one vendor marked it as malicious, based on the code reuse, you can immediately determine that this file is malicious and it's from North Korean actor, which is Lazarus in this case. So this is a quick example of how you can use Intezer Analyze and the genetic software mapping technology for attributing and classifying threats within seconds. You can also see here strings which appear in this specific sample and other Lazarus samples, but I can explain it deeply on a different session. Now, the second use case that I wanted to show you is how to analyze an endpoint using this technology. So this could be part of proactive operation, or maybe part of the incident response process. But once you have a suspicious endpoint which you want to investigate, you can ship an executable that we wrote that scans the whole memory image. And for every page with executable privileges, we scan it in our technology. This means that if you have any threat that is running on this specific endpoint, even if it's fully volatile, nothing on this, using our technology, we will be able to detect it. So let's take this example. After executing this scanner on an endpoint, we detected that it's infected. Although we see some trusted modules here from Microsoft, we can also detect this Emotet code loaded in memory. So we see some Packer genes here, but we also see the Emotet genes. This means that this endpoint is infected with Emotet. Maybe it has no malicious payload on disk. Some Emotet payloads are like that. But still, we were able to detect it using the genetic software mapping. I think this is extremely useful for government certs and large certs that has a lot of operations going on. So instead of creating copies of the hard drives in the network they need to analyze, they can just ship this executable, execute it, and after a few minutes, you can see the output of this endpoint without full memory dump, without media, without need to copy the whole media back to your lab and so on. Yeah, this is a game changer for national certs and certs that are doing large scale investigations. All right, we will also focus obviously on the code connections cluster soon, so I'll skip on that. And we can also accelerate reverse engineering process and doing threat hunting based on code. The accelerate reverse engineering process is based on our IDA Pro plugin we have, which basically gives you all the insights you need within IDA. So you get the genetic software mapping insights without leaving the IDA's interface. And of course, if you're interested in that, I can explain more during the Q&A session. Please let me know. And we can also do threat hunting based on code, which I think is very interesting for national search, since, as I mentioned, doing threat hunting based on strings or based on IPs, it's not that effective. It's relatively easy for the attackers to modify and change those IOCs, but once you base it on code, you make it also impossible for the attackers to change it. So since we know which part of the code is malicious and which part of the code is trusted, we can base and build YARA rules only from the malicious code. And the YARA rules are code-based, meaning that every time we would see this code from Lazarus again, you could get a notification. So again, that's not the main focus of the session, but this is indeed something that you can and should do using the technology. This is how we roll in the research team. We find new threats based on that. And in our reputation, we were able to detect Chinese APTs, North Korean ones, Russian ones, Iranian ones, and it's all based on this threat hunting method. But yet again, we're here to focus on the code connections cluster. Yeah, and this is the main focus. So why is it even important to build code connections cluster? How can this be useful for your needs? So first of all, it can help you accelerate the investigation during an incident. Once you can correlate few files together based on the code, if you investigate one sample, you can conclude and share this conclusion with the other samples in the cluster as well. For example, you have 5 files that are 99% identical to each other based on the assembly. If you investigate one of them and it's trusted, the others are trusted, probably trusted as well. Same for malicious. So this could save you a lot of time during handling an incident. And as you all know, time is a very expensive resource during incidents. You can also use the cluster for attributing samples. If you have a cluster of samples, which you know that they are APTX, and now you have a new sample that share the same, that shares code with this specific cluster, and it has strong connections with it, this probably means you can attribute this specific sample to this cluster. It requires further digging, of course, but it's a great lead. Now, track variants of known samples. This is something, and also monitor groups and actors. This is something that I'll show you right away with the example of the code connections cluster. So this whole session came to our mind after we did this Russian APT ecosystem map. We released it a few months ago, and actually it was a great success, and was the result of a long research we've done for months in our research team. Now, we wanted to map all the Russian APT groups that are active in the past few years, and to find the connections and build clusters between all those groups. So the research team at Intezer, along with the research team at Checkpoint, we collected a lot of samples together. We opened each and every report regarding Russian actors by security vendors and national certs in the past, I would say 20 years. We run all those samples that we've collected in Intezer Analyze engine, the code reuse engine I just showed you, to see the connections between the different groups. Then we analyzed the connections and we presented it on them on this graph. Now, this graph is interactive. Of course, you can zoom in, you can zoom out. And we had quite interesting conclusions, actually. Every node that you see here on this graph is a tool. You can see the name of the tool, the version, and once you over your mouse on this specific node, you can see the connections to other tools and families. Now, you see here the different colors here. Different colors represents different actors. So immediately you can see this cluster here, right? Or you can spot this cluster over here. And once you zoom in, you can see that all of those samples are black energy samples, right? And they share code with each other based on the assembly, based only on the assembly instructions and the capabilities of the file. So all the black energy samples share code with each other. Now, this is very interesting because you can track black energy samples using this graph, right? Once you see a new sample that is correlated with all those nodes, it probably means it's a new black energy sample. Now, moreover, you can see that this black energy module of password stealing shares some code with this specific pinch juke tool from APT 29. So you can also conclude technical intelligence, using this specific graph. Now, this graph is just a use case of what you can achieve using this technology. By the way, once you click on a specific node, you can get further information about it. So, for example, we can see here, once I click on that, I get the short description of OBOS. I can further read about it, and of course, get links to the genetic software mapping report, which we base the whole map on this technology we've developed at Intezer. So this is extremely useful, and by the end of this workshop, you guys will be able to create the same map, but for your own specific needs. So, for example, if you have your own actors that you're tracing, and you want to build this relation graph, or if I'll go back to the use cases, if you want to analyze quickly a specific cluster you built and analyze just one file instead of analyzing the whole files all the files in this cluster to save time during an investigation, you will be able to do that as well. By the way, the Russian APT Ecosystem Map is open source, both the raw data and the GUI. We also released Yara rules based on the connections we've detected. So I invite you all to try. So what you should do in order to create this graph by yourself. And this is, I think it's the last slide before I show you a live demo. So first of all, you need to collect samples, right? It depends on your resources. You can use Intezer Analyze platform to collect those samples as well, but you need to have the binaries or hashes of those binaries. Then you need to execute the clustering script, which is available on our GitHub page. This is exactly the same script I'm going to execute in a few minutes to show you how to build a graph. And then you need to analyze it. Obviously, you can use Gephi for it, which is a tool to analyze graphs. You can build your own web interface. You can use what we built in the Russian APT ecosystem to analyze the data. But you do need to have a platform to analyze the cluster. And once again, the clustering script is available in our GitHub page, and this is the address. And I will also share it at the end of the presentation in the chat. All right, I think it's demo time. Okay, so first of all, this is the GitHub repository where you can download the clustering script. It's our repository, obviously, and please make sure once you use it to update your API key from Intezer Analyze accordingly. This is the only change you need to do before executing the script and generating the graph. Now, I cloned this project. Here it is. And I also gathered, as I said, a list of samples, which I want to build my connection graph based on those samples. I want those samples to be the nodes in the graph. Now, all we need to do is to execute the cluster directory.py script on the samples, this folder of samples. And meanwhile, we let it run. And during the time that the script is running, you can start downloading Gephi, which is an open source platform to analyze graphs. I'm going to open the graph that is being generated right now using the script with Gephi and start analyzing it based on that. One more feature I wanted to show you in Intezer Analyze, and it also relates to the way I collected the samples, is that we have a search malware family feature in our genome database. And the samples I'm executing right now is just basically a list of the Lazarus samples that exist in our genome database for the past 6 months. So what's interesting is that you can just search here Lazarus and get all the Lazarus samples that we have in our genome database, including short description of the family. And then since you're using the platform, you can also download those specific samples. It takes a few seconds to collect everything, but meanwhile, oh, here it is. You can see here all the samples that we have, or a few of the samples that we have, not all of them actually, in our genome database based on the code, including the first time we've seen those specific samples. And what I did is that I took the top 50 samples, put them all in the same folder, and yeah, now I'm basically analyzing all those samples in order to create a connection graph. The search family feature is extremely useful, especially if you have a lead for a specific family and you want to investigate more. So let's say that you heard on the news that APT 29 is on the rise. You can click here, get some further information about the family. For example, synonyms. As you know, different malware vendors are using different names for the same family sometimes. You can get the references here for further reading. and of course, see the actual samples that share code or relates to this specific family. And of course, once you click on a specific family, on a specific sample, you get the genetic analysis of it. And this is the web interface that I showed you before. It's very straightforward. This one, for example, is a Linux file, L file, which is statically linked. And as you can see, it's APT 29. So this is just another cool feature that you can use our platform, that exists in our platform. And it's very unique, right? To search family and see the samples that has code and relates to this specific family. And this is how I collected the samples for this specific demo. Okay, and now we see that the graph was saved, which means that the script is now done and we can start analyzing the graph. So for that, as I said, we should use Gephi. This is Gephi's interface. And first thing we need to do is to load the graph we just created. So to do so, we should go to File, Open. Here it is, and I have this output.gexif. Let's load it. And this is how our graph looks like at the first place. Well, this is basically very hard to understand, right? It's not clear what is going on, where are all the clusters, and so on and so forth. But don't worry, we will make the graph prettier. I just want quickly to go over the tabs here. So we have the overview, which is the tab that we are in right now, the data laboratory, where basically you can see the raw data of this specific graph. So you can see here all the labels that the nodes that we have, and each node has an ID, and of course the label. And we can also see the edges between the different nodes, source and target, which means where the connection goes from and to. This graph is not direct graph, so it doesn't mean much which node would be the source and which node would be the target, but just in case. And of course, we see here also the gene count of the shared code between the different samples. And lastly, preview. After generating the graph, we can see it here. And this is how the graph looks like originally, right? It looks like a mess, but that's not the case, obviously. So we'll go back to overview, where we can actually work with the graph. And besides doing some cosmetic changes like changing the colors of the node and adding different labels to this graph, we can also execute some algorithms on the graph layout, right? So there are a few algorithms here which you can execute. I like to execute, first of all, false atlas. That's how it looks like. Then we need to stop it. And then this one. And suddenly the graph looks much better, right? You can see here immediately now we have clusters. You see this one here and this large cluster over here. And yeah, just a reminder, each and every node here in this graph is a sample based on the sample list I just showed you here, okay? So this looks good, right? Some of the clusters already appear here. I can also stop the algorithms. And now we'll go back to preview and regenerate the graph. So this is how the graph looks like right now. I can zoom in, zoom out. This is good. It's interesting, and it gets better and better. Now, for example, if I'll go back to the use cases we said that are relevant for the graph, all those samples that are in the same cluster share code with each other, right? That's why they are all on the same cluster. So if you're investigating those files and those files are suspicious, what you can do, instead of analyzing each and every one of those files, you can just spend time on analyzing one file and then share your conclusion regarding this file and assume that it's relevant for all the files here. And again, the connections are based on the assembly instructions. It's not based on hash, string, or things that are easy for the attackers to change. It's actually the actual capabilities that this file has. That's why it's so powerful, right? That's what makes it so powerful. Now, how do you continue from here? Okay, so first thing we need to add is the labels, right? Because we don't know what is going on with each node. So here under the settings of the preview, you can just add here show labels. Now let's refresh, and suddenly we get all this text, right? It's not that comfortable, but we're going there. So we can also change here the font size, right? And then don't forget to make sure you click refresh. So now it's a bit more comfortable, right? I can even change it, makes it a bit smaller. And we can start analyzing here the samples. So I see this one, 7EF72. All right, so I know that I should go back here to the folder of files and search for 7EF72, right? And analyzing this chosen candidate from this specific graph. Now, if you have a sample which you suspect is related to a malware family or a specific actor, you can also use this graph to determine that. All you need to do is to load this specific sample using the script I just showed you to the existing graph you have of this actor or nation state. It depends on the raw data you have. And then if you see this new sample relates to this specific cluster, it means that they share code, right? Now, basically, this is what we do with Intezer Analyze. But if you want to see it in a more visual way or on a graph, this is also possible using the way I just described. So as you can see, this feature is extremely useful for threat intelligence, for malware analysis, and to do forensics and accelerate the investigation process. Now, what's interesting here is that you can do it based on your own samples, right? As Intezer Analyze customers, you do not have to share the samples with us. You can, if you have a cluster, if you have samples that are private, which means that they do not exist in VirusTotal or not in any other place online, you can also build this relations graph using our technology. So it's kind of like you can bring your own samples. It doesn't have to be based on samples that we have in Intezer Analyze. And I'll be happy to explain more on that if there are any questions. Now, the next step in analyzing this graph is either choosing the samples here and go back to IDA or to Intezer Analyze for further investigation, or if you want to make this graph interactive, because as you see here, it's not clickable, right? It's based on Gephi. You can use the web GUI we built in the Russian APT map that, as I said, is open source to do it in a more interactive way, which is clickable, and you get further information per sample, and so on and so forth. Well, yeah, so this is basically it. I'll be happy to answer any question you may have. And anyway, thank you very much for your time. And I hope you enjoyed and you find this workshop useful. Please, if you have any questions, let me know right now. Thank you.